So I'm very happy that I'm here. This is, I don't know, the seventh or eighth ICMS. Um, yeah, great. So, as the chairperson did say, it's my added responsibility to keep you entertained so that you guys don't sleep. So I'll be glancing at you guys once in a while. If somebody is closing his or her eye, I will be really, really staring at that person continuously. And okay, till that person opens the eye. So that's the kind of thing. So see that your eyes are open. Have you seen Mr. Bean by any chance? You know how Mr. Bean keeps eyes open? So do something so that the eyes are open. And this talk is for the students. And I'm aware that there are BSc students, BSc level students, and MSc students. So there is nothing that is going to be very, very high tech. If at all there is something high tech, I will bring it to the lowest tech possible so that we can have a two-way communication. And I would want to stop at 35 minutes so that there will be real handsome 10 minutes for you to ask me questions and for me to answer your questions. And in case you don't ask any question, I won't be very, very happy about it. I would think there was something wrong with my communication skill. I will try to improve it next time. So, okay, let's get going. I heard about cells, and this is cell signaling. Signaling comes later. I heard about cells when I was in school, maybe in the 6th grade, 7th grade. And all the time in my life, I had, school time, the college time, I had a lady teacher teaching biology. Those days, men would never teach biology as if, it's a bad subject. So I also had this female teacher teaching me biology, talking about cells. She told me that we are made of cells. Okay, you believe it. And I had never even seen the cells in reality. Textbook is there. Textbook has a picture of the cell. I also put it over here. Animals have cells. Plants also have cells. Some differences, lot of similarities. And you keep believing it and you keep writing answers to the questions and you pass. You all passed, I also passed. 6th, 7th, 8th. And then one day this lady, my teacher, talked about cells dividing. So there is this mummy cell. Mummy cell will be eating a lot of food, will be sleeping, and then one day she will divide to make 2 baby cells. Similar to the mummy cell. Mummy cell is gone, now you have 2 baby cells. So poor me, I asked, okay, well, who told this cell to divide? I have not seen my mother dividing to make 2 mummies, yeah? But this cell is dividing. She told me to sit down. So I did sit down for one moment. I said, but you answer the question, I will sit down. So she told me one more round that okay, this is not there in the syllabus. Like who told this cell to divide into 2 was never written in the textbook. So why should she teach? It was written that the cells divide in the textbook and she taught that much. But who told you is the question, that can come only from a stupid guy like me. She sent me to the principal's room. Normally that's the solution, when you're really, really problematic in the class, you reach the principal's room. I told that guy, I asked this lady a question. The lady said, no. Now the principal asked, what was the question? I told, this is the question. The principal guy who is an MA English, how will he know what is cell division? So he told, go to the class, don't create problem. Fine. So that time went. Then came, so this was the question that I asked, how do cells divide? Anyway, I thought that was an important question. They all thought it's a trivial question. Later, these 3 guys understood and cracked cell division, mitosis. And they got Nobel Prize. It was not that they got something very, very small. And several years after, I asked this question to my teacher. I asked this question to my teacher in 1970s, mid-70s. And they bagged a Nobel Prize in 2001. I had the question in 1970. I lost Nobel Prize, the first Nobel Prize actually, right? In case this lady had permitted me to do some experiments over there, if she had cooperated, if my principal was decent, I would have got this Nobel Prize, right? And I didn't get it. These guys got it several years later, 20 years later. I have to feel sad about it, right? And I felt very sad about it. Anyway, that's okay. Then she talked about meiosis. That meiosis happens. So all these cells divide into 2, normally through mitosis, and you have 2 baby cells, as I said, identical baby cells. Now meiosis is something different. There are some cells, they will divide through mitosis and all that. Then comes a day they will divide through meiosis. They will make two baby cells, ultimately 4 baby cells. They are not like the mother cell. They have only half the load of DNA. So if 2 and 23 pair is a number, our chromosome number, through meiosis you only get 23 chromosomes. So you get 23 chromosomes from your father, because my father knew to make these cells. And 23 cells from my mother, she also knew how to do it, though I did not know how to do it. Now I asked this lady, why is it that, so she told me that this happens only in some tissues, in testis and ovary. I knew where the testis is, but I had no idea where the ovary is. I believed that the ovary is there somewhere. There was picture, it was somewhere deep inside. I never explored where it is, but the testis I knew is there. And I asked her, why is it that meiosis happens only in the testis? Oh, I won't talk about the ovary because it's a kind of clumsy meiosis there. So this is better thing to talk about meiosis. So it happens a little differently in ovary. If you ask me later, I will tell, but in public I won't tell. So the question was, okay, how is it that these cells are dividing through meiosis now? And why is it that it's happening in the testis and also in the ovary? Again, not there in the textbook. Textbook, it was there that, okay, meiosis happens in the testis, nothing, or in the ovary, nothing more than that. Why is it that it did not happen on my finger? It was not there. Now I thought, well, this is a problem. These cells in the testis can divide through meiosis, but my finger cells, skin cells don't divide through meiosis. I reached the principal one more round, and same story. I was told to go and sit quietly in the class. So this second question remained with me all the time. I thought by any chance, if I get a chance to do some science, I will do it. That was the thing. And mitosis also was there with me at that time as a question. And then comes this fertilization. One cell from the father, one cell from the mother, they fuse together to make baby. No big deal. Now, when you grow up, when you are in school and all that, you see that there are some ladies around, maybe in your neighborhood, your relatives, some ladies' tummies swell up. After some time, they have a baby. So I knew babies are there in the tummy. Now the question was, how did the baby get into the tummy? So I had no clue. Those days, this Google and ChatGPT and all were not there. You had to ask your father and the mother. So I asked my mother, how is it that this stuff gets into your tummy? And my mother had these marks also. So I knew that it was there. And I was there. So I asked her, how does it happen? She told, ask your father. So I went to him. I asked him, how did that happen? She told, when you grow up a little big, you will understand all those things. Now, I was growing from the 7th grade. I got into the 8th grade. I asked her, well, can you tell me? He told me, you have to grow further. So 8th, 9th, 10th, 11th, 12th, then I started knowing a bit about what all things happen. Anyway, that happened. And when I was talking to school kids in between, I tried to tell about this fertilization, like daddy cells, mommy cell fusing together. So it's a group of school kids visiting the Rajiv Gandhi Center, and we had those kinds of open houses. So the kids are there in my cabin, and my lab was there. So normally I used to call the kids to my cabin, then I give a little talk. Then I would let them loose in the laboratory. That's the way I used to do. And my lab people will take care of the rest of the things. So I gave this little introductory talk that this is the way it happens. I work on fertilization. This cell, they fuse together to make a baby cell, and then the baby cell grows, and that made you all. So one boy told, Uncle, we know all those things, but how did this daddy cell get into mommy? I told, you have to grow a little big. Then you will also know that. So I think that boy is still waiting, I believe, or maybe he has already grown that much that he knows how his daddy cell got into the mommy cell. Anyway, that was not a major problem. That problem was there, which I understood later. You guys also know how that happens. But beyond that, the point was this, that, okay, before that, let me tell you. So the male gamete, the sperm, is a haploid cell. It's a terminally differentiated cell. They don't do anything beyond their formation. They die much of the time. Same is true with the oocyte also. They also are terminally differentiated. So if you look at them, in isolation, they don't have any future. Sperm cell dies, the oocyte dies. But if you fuse them together, then we have all creatures like me and you, right? But this is possible in situation where you have two oocytes coming into picture, maybe one, maybe two, maybe a little more than that in some cases. So two lucky oocytes and two lucky sperm in a couple's life possibly, making babies. Rest of them, the male guy, today morning Subir was telling 200 million sperm we produce, as if we have no other job to do. Like morning you get up, you make 200 million sperm. And women, they don't do that. They make one oocyte per cycle in normal situation. All our time and energy goes in making sperm. That's why we don't score that good, during studies, because we are busy making sperm. 200 million will be needed by the evening, right? So we are busy with that. That part is okay. But then these two things, haploid cells with no capability to do anything, when they fuse together, you get a zygote. The zygote is totipotent. These two cells had no potency of any given kind. Let me say that I call them nullipotent. They are not dividing cells. Their fate is death. And every day it's happening. And if you fuse them together, we have amazing creatures like us. Each individual is really, really amazing. Now my question was, how do these two cells fuse? I visited the principal one more round. I told this man that we need to fire this lady, my biology teacher. We need a good biology teacher who can teach biology properly. Because this lady, whatever question I asked, she doesn't know. And I thought, okay, I will have an answer to all these things one day, and I will go and meet her to tell her, like, lady, this is the way mitosis happens, this is the way meiosis happens, and this is the way cells fuse. Anyway, the third question, the second question about meiosis, in between, let me tell you, keep it in your mind. The first question I told you, those guys got Nobel Prize. Second question, I did not tell anything about it right now. The third question, how do cells fuse? Then these guys, it's all written who they are. You can identify them. They took a B cell and they took a myeloma cell, they fused it. So they could fuse it. And for fusing those two cells to produce whatever, they got a Nobel Prize in 1984. I had this question, how cells fuse, in 1974. Now, my second Nobel Prize also is gone. In case I had a lab and I had decent situation around, I would have sometimes cracked this. So, I lost the second Nobel Prize, okay? That's all right. So, and then these cells, the middle cell, the greenish or bluish or whatever you see, I don't know. I see it as a little bluish color. And my daughter will tell, I have no idea about the color, because we see whatever number of colors. Females see, I don't know whatever number of colors. 16 million, I don't know, 24 million. I know red, blue, green, and the primary colors I know. But if they say that blue 201 or 203, I had no clue. They have numbers for these colors. I don't know which number this is. This will qualify. But whatever is that color, that is an undifferentiated cell. Call it totipotent. It's a zygote. That makes everything in your body. All the cells that you have in your body came from one single cell. That is amazing. How is that possible? So two cells with no potency fused together to make one single cell which had all the given potencies. You don't have that kind of cell in your body ever. You lost it because that's what made you. You don't have it in reserve. You have some cells which have some level of potency, but you don't have even a single cell in your body which is totipotent. You only had that one single cell which made you. The moment it made you, that is gone. So this cell can make every single thing. Now, that was another question. How can that happen? You say that these cells did not have potency. Now you say that these cells have all the potency. You say that this made every single cell. How can that happen? Anyway, again, same story. We know all these terms. Unipotent means one potency. My skin cell is unipotent. Then you have multipotent. You may have a couple of potencies. Then you may have pluripotency, a little more number of possibilities. Then you have this totipotent cell. So this potency level comes down from totipotency to unipotency. Let me take a pointer here. Okay. All right. Let it work like that. Yeah, so unipotency means that's the lowest level of potency, and it goes up like this, unipotency, totipotency. And you got a sense of it. You must be knowing that as well. And taking a going back. So taking a cell, call it a unipotent cell, if you convert the unipotent cell into a pluripotent cell, not a totipotent cell, these guys grabbed a Nobel Prize. That too, several years later, 2012. So that question as to how this one cell makes all those kinds of cells was a thing that was not known. These guys converted a fibroblast into an induced pluripotent cell which can make several types of cells. So a third Nobel Prize also is gone. So this is the induced pluripotent stem cells. And I did talk about this fusion. And okay, this also I have already told. I think it's a repeat of the slide. So now let me come to the major point that I wanted to tell you. A lot of people cannot make the sperm. That does mean that the meiosis doesn't happen in many people's testes. A lot of people are there. 15% of the people have problem in making a baby. So somewhere or other there is a problem. Some people cannot make the sperm. Some people can make the sperm. The sperm may not be that great looking. Some people may have the sperm that cannot bind to the oocyte. A lot of issues could be there. So they are all coming in this infertility class. So there are various reasons. The female factor is not a thing that I will talk about. I will only talk about the male factor. So I have listed many of the things. So there could be a problem with the spermatogenesis, no sperm, because there are no stem cells in the testis that would ultimately make the sperm. That's a possibility. And there is no division of the spermatogonial stem cells, and meiosis could be failing. And after it all happens in the testis, today morning I think somebody was talking, there is, the sperm undergoes a lot of changes in the epididymis. There was a talk about epididymitis, and I think the first day Shravani talked about epididymis and capacity. A lot of changes happen to the sperm during post-testicular development outside the testis. And then fertilization, too many steps are there. Any failure anywhere can create problem. So if you just try to look at it from top to bottom, cumulus, those cells around here, if you can see the pointer, so the sperm has to first go and touch these cells, disperse these cells, and then just come in and in. Then this peachish cover that you see, the sperm has to bind to that, penetrate through that, get into the space, bind to the ulemma, and then get into the oocyte. And then the sperm has to activate the oocyte. Otherwise, oocyte was not a dividing cell, I told you. So once the sperm gets in, the oocyte starts, decides to divide, starts making changes inside to make it capable of dividing. And that's the time where all that reprogramming that I was talking about, how this cell, which is nullipotent, becomes totipotent. That means the sperm enters and the oocyte gets reprogrammed in the presence of the sperm, DNA and other materials, and it makes a cell which becomes totipotent. I normally make an argument. If this finally becomes a zygote, it divides into two, the first division, two blastomeres are there. So if you separate these two blastomeres, in natural situation, it might happen, you will get two babies. But these two babies will be identical because it's from the same zygote, the same DNA load, genome content is there, so they will be identical, right? Not that this one will make half a baby and this one will make half a baby. That won't happen. They can make full babies. That would mean that before the first division, this cell became totipotent. Otherwise, it wouldn't have happened. If it had not become totipotent before the first division, there would have been problem with the development. So both the cells became totipotent before the first division. First division happens in 24 hours after fertilization. So logically speaking, the reprogramming of the cell, the oocyte into a zygote, happened within 24 hours. So you have just 24 hours to study what happened in the oocyte. Take the oocyte, take the sperm, push the sperm into the oocyte, turn on the clock, and look at what all things happened. You can read it out on the 24th hour. You can come down and down and down all the way to zero time if you want to. So we did some studies on that, wasted a lot of time and wasted a lot of money. And we know some things which are happening, but they are really fast reactions. So if anything is wrong over there where the oocyte doesn't get converted into zygote, or if the zygote doesn't get reprogrammed properly, again, ultimately the readout will be in a normal situation, the readout would be that there is infertility, right? That will be the normal readout in a regular life. So if male has the problem, I told you that I won't talk about the female part. So the doctor will catch the guy, tell him to give some semen, and the doctor will do all the semen analysis. So normal volume is 1.5 milliliter, and the doctor will tell, oh, you are not producing 1.5 milliliter semen, as if you produce 1.5 milliliter, he's going to be amazing. If it's little less than 1.5 milliliter, that's the problem. That's actually not the problem. He produces 1 milliliter of semen. How can you say that that is the cause of infertility? But that is one explanation the doctor gives. And the sperm number, the doctor will say minimum 39 million sperm you should produce. Again, as if you make 38 million sperm, you fail. It's like your examination, You pass and fail. You get 40 marks, you pass, you get 39 mark. Then I think you will go and ask for the mercy of the vice chancellor to pass. So if this guy gets 38 million sperm, he has to go and tell, please make me fertile. I have 1 million less. That also cannot happen. And if the sperm concentration goes below 15 million, then the doctor will tell, hey, you have a problem called oligozoospermia. Very nice name for the disease. And the guy comes out and says, hey, I'm oligozoospermic, as if he got an award. And the motility should be 40%. 40% of the sperm should be really, really moving. Again, if 38% moves, he's infertile, okay, according to the doctor, not according to me. And coming down to the morphology, how the sperm looks, 4% should be very handsome. Should be like Salman Khan or Amir Khan and all that, should not be like Johnny Liver or whatever, okay. So, and in case, the second is the case, they are arsenozoospermic, they are all drunk, they don't really move. And if the last one is the case, teratozoospermic. Some guys have a combination of all the things, then the doctor will tell, you are oligo-arseno-teratozoospermic, as if the degree is very big. Like you have BSc, MSc, PhD, this, you get a real long title to the thing. So these are the things doctor would be saying, and the doctor will do whatever kind of thing. And the doctor will make the sperm swim up and up and up. Like take the sperm, put the sperm, at the bottom of the pool, and then all the sperm will be climbing up and up. The ones which are coming up, he will be catching it. Like you do the fishing. The good ones are coming up. So, it's in a test tube. They will have a percol, some high density material. You will put the sperm at the bottom, and you try to look for the sperm at the top, thinking that they are the strongest ones. And using that sperm, they can artificially inseminate the lady by the husband. Even if the husband was able to inseminate the lady, doctor will take a little catheter and do the job for the husband. But you have to pay the money. Husband was doing it free, but here you have to pay the lot of money. And then you have intrauterine insemination. So the sperm will not be put normally at the normal place. Doctor will take the sperm all the way into the uterus. And husband won't be able to do that anyway. So we call it IUI. And then we have gamete intrafallopian tube transfer. Take the sperm, take the oocyte, fertilize it, take the gamete, put it into the fallopian tube. That is called gift. When IVF ET, you make the embryo also inside the petri dish, take the embryo and put it onto the uterus. It will attach, it should attach. And and in case the sperm doesn't bind to the oocyte at all, in a normal situation, what happens when you do all these things, IVF ET, you take the sperm, you take the oocyte, put them together, lot of sperm and one oocyte, put them together, incubate, and fertilization will happen naturally. Then put that thing, at wherever. And if that doesn't work, if the sperm doesn't bind to the oocyte, then what they do is they do what's called ICSI, this one. So ICSI means you take the sperm in a microinjector, inject it into the oocyte. How will you not go into the oocyte? It's the question. The doctor will see that the sperm gets into the oocyte, and then if there is no sperm in the semen, they will try to take the sperm from the epididymis, whichever way. They can take the sperm from the testis. The doctor can take it because there is nothing in the semen. But if they're sometimes the epididymis, sometimes the testis. If that also is not there, they will look for a round spermatid. If the spermatozoa is not there, there will be spermatid. The round spermatid also is haploid. Up to primary spermatocyte is all diploid, secondary spermatocyte onwards it's haploid. So you need a haploid cell nucleus from the guy, that's it. So they will do what they call as a Rosny. And all those things are done through intracytoplasmic sperm injection, sperm or nuclear injection. And I had been thinking all the time from 90s that this is very descriptive, and doctor does whatever. So he will try protocol number 1, that failed 2, 3, 4 times, then protocol 2, 3, 4, several years go. So we have patients who had been visiting clinics for 10 years. After marriage, still they don't have a baby. 30% cases, there is success. 70% cases, there is failure in these setups. And these things are all very costly, the ones that I was talking about. The range could be somewhere from 5,000 rupees to start with, like artificial insemination by the husband, they charge close to 5,000 bucks. And all the way down to ICSI, it could be somewhere around 1.5 lakh to 2.5 lakhs in the normal market rate. So, and they go through multiple rounds of this process, so you spend quite a lot of money, still 70% people don't have a baby. So we wanted to see why is it happening like that. The sperm is available, or haploid nucleus is available, the oocyte is available, and the doctor can make sometimes an embryo also. Sometimes he cannot make an embryo, and then the baby doesn't come. So the question was, are there problems with the sperm? You have sperm, number could be less, whatever kind of thing, but ultimately you got a haploid nucleus. How doesn't it make a baby? That's the question. So the first thing is the sperm has to bind to the oocyte. Molecules are needed, proteins are needed for the sperm to bind to the oocyte, because sperm is from somebody, oocyte is from somebody else, so there has to be a lot of recognition, molecules are needed. So AlphaFold got Nobel Prize. Using AlphaFold recently, they discovered molecules. 3 molecules on the sperm, like there's a TMM81, Isomo1, and SPACA6. 3 proteins on the sperm at 3 different positions. And you have the oocyte, and this is there both in animal model and in humans also. In zebrafish model, these 3 proteins, if they stay at 3 separate positions, distant positions on the sperm cannot bind to the oocyte. But the moment oocyte is there somewhere in the vicinity, these 3 proteins come together. So they move laterally on the membrane, come together. It can bind to a receptor called bouncer on the oocyte. That's the kind of locking mechanism. And the sperm will get into the oocyte. And if TMM81 is not there, binding will not happen. Same is true for Isomo1 or SPACA6. And on the other side, if bouncer is not there, there will be no binding at all. So 3 proteins on the sperm are needed. They are needed to be present at the right place at the right time. And in human beings, bouncer homolog is there, Juno. So if you don't have Juno, there is a problem. Doctor doesn't see it. And the guys, if they don't have any of the 3 proteins, it's a problem. And even if all the 3 proteins are not there, if they cannot come together, it's a problem. So they won't bind automatically. All the IVF will fail. Sperm will not be able to bind to the oocyte on its own. That's the story. So we thought, okay, and there are many other things that sperm proteins will do. So we thought we will sit down. Well, AlphaFold story came recently only. We started this in the 1990s. So we thought about looking at the sperm proteins in fertile guys versus infertile guys. So we decided to initiate the program in early 90s. So I hired the first student to work on human sperm. Till that time we were working on animal models. So now the question is getting human semen, because you need sperm. So I had one boy. His body weight was 35 kilogram at that time when he joined me for PhD program after qualifying CSIR, JRF. So I told this is the project. We need to have human sperm. So infertile sample is available in the hospital. A lot of patients go over there. So we need to have an understanding with the doctor. You can get the sperm. Normal one, hospital patients, not patients exactly, males are good ones. They are not clinically normal as far as fertility is concerned. So they would not give sperm. Whatever happens, they won't give semen. So what do? Normal semen is not available. Abnormal one, infertile ones are available. They will tell, go do use whatever, because they need a baby. Normal guys, they already have a baby. They are going to the clinic because the lady has a problem, right? So they think we are fine, but we are not going to give you the sperm. So today Subir was telling, if you try to catch the guy and take the testes out, they all will run away. Forget about testes. I never asked for a testis. Later I asked for testis. That comes later only. But I was asking for semen. Nobody will give. So we had a guy, happened to be my wife's student, Malini's student. So he told, hey, there is a trick, boys' hostel is there, a lot of boys are there. We collect it. So how much do you want? I told, how much means what? I don't need liters, I need some samples, 5, 10, 15, 20, whatever number. So he went and started canvassing. So 2-3 people gave semen, and then they fell flat as if they are going to die. As if they are very weak now, they cannot even go to the class because morning they gave semen. And this boy had the answer, hey, we will give them 1 egg and some milk if they give semen. So we collect semen, we give egg and milk. And then the guys jump up and go to classroom. So we started getting normal sample. Abnormal sample anyway we were getting. So once we had that, we bring them to the lab, we take the sperm, lyse it, do whatever, take the protein. How much more minutes I have? 5 minutes. Oh my gosh. So we will do what's called as electrophoresis, 1D, 2D electrophoresis. I'm going to run super fast. And the technique kept on evolving. And so we take the proteins which are there in one sample, normal sample, absent in the infertile sample. 5 minutes for 35 minutes or 45 minutes? Okay. What did you say? Okay, 10 minutes. Okay, yeah, right. So, so we will, yeah. So we run it on 2 different gels, compare the gels, normal sample, abnormal sample, and match the bands. And we have softwares to match it. And finally, it's almost like this digital thing, this Digiyatra and all that. So kind of image-based, mapping. So whatever protein is different, we take it out, we will do what's called as N-terminal sequencing. We will get the N-terminus of the protein, which will correspond to the 5' end of the mRNA. And the second thing we started doing was a dyes. So we will label one set of protein, the normal one with one color, the protein from the abnormal one with another color. Put them together, run it on one lane. So whichever one is red is present in one guy, whichever is in green is present in the other guy, and whichever one is yellowish or orangish is present in both the people. And collect those ones which are either green or red and analyze it using N-terminal sequencing. So you get a couple of amino acids converted into the predicted N-terminal, the 5' sequence, and do RT-PCR to get the genes. Then came LCMS, years went and LCMS came. So we started doing all kinds of MS-based protein identification, whether it's label-free or label-based or whatever. And we have those proteins which are differentially displayed. Fertile ones these are the ones which are present only in the fertile individual. These molecules only in the infertile individuals. Huge number of proteins were there. And don't try to read it. These are all differential displays present. These are in a huge number of patients, somewhere around 3,000 plus patients actually, normal versus infertile. And then we check because many of them have predicted functions or non-functions. We see that, okay, well, if we inhibit this molecule, will it happen? So we have these assays in vitro. We have the oocytes. These are the hamster oocytes. Human sperm can bind to hamster oocytes because you cannot really work on human oocytes. So we will do that assay, the Sona pellucida binding assay. We will neutralize the molecule using antibodies. We generate antibodies and try to see whether the sperm binds to the Sona pellucida or the sperm is failing to bind to the oolemma. We remove the Sona pellucida to see whether the sperm can bind to the oolemma. These things are where the sperm has bound. And in some situations, there is no sperm on the oolemal surface. And whether it can really inhibit the early division of the oocyte. And then you have a bunch of molecules. You have to decide now after that what are you going to do, because I need Nobel Prize, right? So we had a couple of favorite molecules based on some predictions, and I will browse through one molecule which is cyclin M1, because it's a cell cycle regulating molecule-like molecule which we identified. There was a reason it's there in the testis and also in the brain. Why is there in the brain? We don't worry about it. But we were more interested because this molecule has this testicular haploid express gene domain. So I thought, this will possibly make a cell into a haploid cell. So we did a lot of studies focusing on this molecule to see if by regulating this molecule we can push a cell into meiosis. So we looked for its expression. They are there in the germline stem cells, all that green spots that you see. We checked for its expression during the development of the animal. Somewhere around embryonic state 13.5, you see the expression level coming up, and then it stays like that for the rest of the animal's life. And these cells express c-KIT plus c-KIT. So one channel is c-KIT and one channel is cyclin M1. And it also expresses OCT4. So they are germ cells and they are stem cells. We took those cells out, we started culturing it, it made bodies, embryoid body-like bodies, ES-like colonies. So they are integrin alpha positive, GFR alpha positive, and alkaline phosphatase positive. And we removed some of the required reagents inside, like LIF and GDNF and all that. It made bodies which will express nestin, desmin, PDX1, telling that this is like ectodermal, mesodermal, endodermal. So they can make all the three germ layers. They are truly stem cell-like cells. We did a lot of things to overexpress, silence this molecule. Ultimately, after all this 30, 40 years of activity, I should tell you that we could not produce a haploid cell till now. So I have spent quite a good amount of money of the government, published papers, but we could not make a haploid cell. But this my one student, Nomesh, he developed a co-culture, so a Sertoli cell. Subir was talking about Sertoli cell, and we made a Sertoli cell feeder line. On that, you put the germline stem cell. In the entire plate, some colonies made cells like this. The bottommost one is a haploid cell. So we could make some haploid cells, but not in huge quantity. You're sleeping? No. One is lying in the other person's lap. So, okay. So, but it's possible, possibly, because human cells will be more easy to convert stem cells into, other types of cells. And we can possibly make sperm and egg in the laboratory, not in big quantity, but we can make it in case somebody doesn't have a haploid cell at all. We should be able to make a haploid cell for those guys using this technology. And all those proteins that I was talking about, we collected some proteins based on the availability of the antibody and the quality of the antibody available. We made an antibody array, and we started looking at the patients for its presence, and controls also are there. A couple of proteins. I will show a couple of graphs like this about the expression. Green ones are normal, red ones are the infertile guys. So after all that, we say that, there are, zoospermia specific fingerprint pattern. The absence of those molecules in yellow color will indicate there is a major defect. So those patients, if they go for some of the attempts, artificial insemination or whatever kind of assisted reproductive technology trials, it has a very huge chance of failure. So all those things in yellow color, if the patients have those kinds of syndromes, those yellow ones, if those molecules are not present in those guys, the chance is that they won't make a baby using a given protocol. You may have to have another protocol, or maybe none of them will work. So we can tell them, don't waste money, you won't be able to make a baby. It could be discouraging news, but that's the reality. So, yes, pretty much that is what I wanted to tell you. A lot of people did things, and the guy who started collecting semen in my laboratory was this guy, Sharad Purohit. He's the one who was trying to collect semen, and the brilliant individual who told about this one egg and 250 milliliter of milk is not there in my photograph. He was not my student. So he was my neighbor student. Yeah, so a lot of people worked. Yeah, done. Yeah. So everybody funded, and I put this because if I talk about reproduction today, whatever you have, you have to sell it in the market. You need money. Somebody has to give you money, to do the research. So if you look at the funding agencies' trust, you won't see reproduction in many cases. 70s, 80s, 90s, they put a lot of money. and nothing much happened possibly other than publishing papers. So now the fashion in funding is something like this genes to that genes. Many of you, RIP gene also has gone. You have this loose stuff, whatever is the name, I don't know what's the name. So, so in case you want to attract some funds, there are these two additional questions. One thing is the aging, which is there in all funding agencies program. So germ cells, do they age just like your somatic cells? In case they age with the same pace. But one thing I should tell you, the telomerase activity and DNA repair enzymes are robust in the germ cells, the germline stem cells, compared to adult cells. So they are really far better. I have data, or some data also. So there is a reason to look at the somatic cells versus germ cells in their aging profile. That is one place where we can grab funds. And the other one is lot of proteins which are present in the testes, expressed normally in the testes, and testis doesn't develop cancer. Today morning, the speaker was telling about epididymal cancer, 0.02 or 0.02 or whatever. Testicle cancer also is somewhere around 0.05%. But 250 antigens are expressed in the testis which are associated with cancer. So these testis-specific molecules, if they are there in some other tissue, say breast or ovary or whatever, there will be cancer. So we call it as a CTA, cancer testis antigens. So what these CTAs do in the testis, why is it that they make everything go wrong in other cells which normally don't express that? It's another amazing area where you can possibly generate funds. Thank you.
Thank you. Thank you very much. One question, because the 45 minutes over. One question. Already 15 minutes late, so please one question, yeah.
Yeah, sir, fantastic talk. So I was just wondering, this cycling M1 is present in the SSCs and also in the differentiated spermatocytes. So, like ID4, the level decreases as you differentiate. So what about the levels of this cycling M1? Does it, is it stochastic?
What's the level of?
Cycling M1.
During development?
Yeah, in the SSC versus differentiated.
Oh, okay. Eventually their quantity comes far down, almost undetectable in the later phases. But there is a speck available, visible on the sperm, but it's very, very, very less. But that may not have a function, at least in the germ cells, in the sperm, because it has nothing to do with anything on the sperm at that stage, because sperm is not transcriptionally or translationally active, right? But in the beginning, yes, huge amount is there. If you see that, remember that image, the outer layer cells are really, really green in color.
Thank you. Thank you very much. Thank you.
Okay, thank you.
Thanks a lot. Yeah, I think something